Right. Okay. So we shall start with today's session on DNA fingerprinting, DNA barcoding, and applications in the biotech industry. Before I start with the next one, the study material was sent to Dr. Manjunath Ma'am, and I hope some of you might have looked into some of the the research publications which were provided. And if not, never mind. We can start today with some introduction. And today we shall be starting with the DNA barcoding. And this is going to be the no. Please, if my there's a problem with my, again, like I said yesterday, there's a problem with my my the volume of the voice, or there's a problem with the flow of the talk, whether it's on the higher side or on the lower side. Kindly do let me know. And if you have any questions, please feel free to unmute your mic and ask questions right there. The for the talk today, the slide plan is going to be like this. We'll start with the principle of DNA barcoding. We'll start with the workflow and all the steps of DNA barcoding, but there's something which I haven't included in the content section because I built it up a bit later, and that is DNA barcoding goes basically this is meant for species identification, not species identification but plant identification, and finding out which species the plant belongs to. And that is very closely tied up with the concept of species. So what DNA barcoding can do and what it cannot do, both of these things are important. So so before we give me a second, give me 1 second. Let me see if I got the correct slide. Okay. So let me first go to what DNA barcoding is. Now, what is DNA barcoding? This was this concept was put forth by scientists from Cambridge University, and also later he moved to University of Gulf Canada, Dr. Paul Herbert. So what Paul Herbert found out was that from very small pieces of DNA, fragments of DNA, if we can find out that sequence, and from that we can do the complete plant identification. So what that basically means is, what he said was, DNA barcoding is microgenomic identification system. Microgenomic identification system. So that means even from a very small piece of DNA, if you can sequence that and find out what that sequence is, you don't need to look at either the morphology or even the whole of genome. Just that little bit of DNA, just a tiny fragment of the DNA, can permit the life's discrimination through the analysis of a small segment of DNA. So, and then underneath what I've written is, this DNA, small fragment of DNA, this can be from mitochondria, plastids, ribosomes, nuclear genomes, and so that's how it is. So, and let's continue to find out a bit further about it. On the upper part, you see this is an orchid, and on the lower part, you see the flower, the magnolia, both of these flowers, and on the right hand side, you see the DNA barcodes. And if we go lower down, you can see, and DNA barcoding is good for not just plants but also for animals and the microorganisms. In fact, this is a bit more successful in microorganisms and animals than it is in plants. Anyway, so from the life forms, we extract DNA as we discussed yesterday, and then from there on we can we need to do the DNA barcoding, which we shall be discussing there. Now, before we go ahead, this is this is a plant which I worked on in Malaysia. This is pak choy, and these days we all know pak choy is becoming it's quite In India, this is, I mean, although in the southeast it's been consumed for a long time, but in India it's only now coming up, and it's being presented more like an exotic vegetable. And it's a shame that it's only now that we are beginning to eat it, and it's such a lovely plant. It's a leafy vegetable, and you can see the diversity. It's a really diverse sort of genus vegetable, and well, this is a, and you can also see on the lower part Greenworld Genetics. That was the company where I was working. Okay, and these are some of the barcodes which I developed. So I mean, and we will go further on it what exactly these sequences are doing. But all these sequences are DNA barcodes, and on the basis of this we can identify the plants. But let's go a bit further down to make more sense of it. Like I said, while we are discussing DNA barcoding, there are a lot of other things which we shall have to discuss along the way. And one of the things that we skipped yesterday, and now is the time to discuss, and that is how do we compare and contrast DNA fingerprinting and DNA barcoding? Because some of the people, for some of the people, especially the beginners, this could be a bit confusing. Now, when we talk about DNA barcoding, we are talking about higher level discrimination. So normally DNA barcoding is good for looking at the phylum level, order level, genus level, and the species level. So at the higher levels. DNA fingerprinting is at the lower level. Lower level means variety within a species or species within a genus. So we use DNA fingerprinting normally for varieties within a species or species within a genus, but not beyond genus. If you want to go beyond genus, you really have to do DNA barcoding. And secondly, the concept is very different. You see, in DNA barcoding, what we do is, as we will discuss, in the last slide, like you saw, there was a sequence. There was there were 4 or 5 DNA sequences which I presented from my own work, and those sequences, they are blasted against a database. So the people somewhere, they have already identified the plants. From those plants, once it's been identified, they extract the DNA, they sequence those bits of DNA fragments, and they say, okay, we have identified it. This is this particular plant, okay, let's say pak choi, and they put it back into the database, which is accessible to everybody. And they say that, okay, here is the sequence, and we found that from this sequence the plant is this. So that database begins to build up. So let's say if 1,000 or 1,000,000 people do a lot of identifications, and they are confirmed that yes, our plant was pak choi, they will say, okay, we know that this was pak choi and this is the sequence, and we are 100% sure, or we are totally sure, and they will put it back and they will give it to the database that this is actually pak choi. And the next time somebody is going across any plant they want to identify, they will take a leaf section, they will extract DNA, they will look at those part of DNA from that segment, and they will feed that into the database. They will compare it with the database, and the database will say, well, we have seen this sequence before, and this sequence belongs to this plant, and this is the species. And that is how the identification is done. So the bottom line is, for DNA barcoding, we need a database. So for DNA barcoding, we need a database against which the DNA sequence from our plant is compared. In case of DNA fingerprinting, we're not comparing it with anybody. We simply create our own DNA fingerprints, and we say, okay, well, this is our fingerprint. If somebody wants to cross-check it, you can cross-check it, because we are not identifying. We are simply telling. It's not. So in that case, we don't need any DNA database. So these are the two. That is the difference between fingerprinting and barcoding. Right. Now, before we go further into DNA barcoding, we really need to go into the concept of species. Here is a very old diagram which some of you might have already seen. Now, this is the tree of life. And many of us are already aware that all life forms are basically interconnected. We all started from a certain source, and then slowly again probably starting with the whatever life forms, the unicellular, and then the fishes, and then all the way, and slowly, the plants and animals and the higher forms, they emerged. So whole of life the whole of life, it's they are all interconnected, and this is it becomes a tree of life. Really, it's all connected. Now here is another just to say that this is 1834, so obviously this concept has been there for quite a bit of time, and this knowledge is already there, that whole of life in this on this earth is interconnected. I would like to this interconnection is important from the DNA barcoding and the species point of view, which will be a bit more clear when we go to the next slide. Now here, in this case, this is this is hang on, this is a crop. Okay, this is one. This is some crop here, and what this picture shows is, diagram shows is, if you look at the right hand side, what we see is, I hope my cursor is available. Could somebody please let me know, when I move the cursor, can the cursor be seen on the screen for you guys? Please, if you could let me know. Can the cursor be seen? When I move the cursor, can you see the cursor? Could somebody unmute and let me know, please? Hello. Is the cursor visible? Kindly unmute and let me know. Hello. 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 Is the cursor is the cursor visible? Okay, thank you. So in this particular case, we can say this black blob, this is a germplasm. And these circles, like the smaller circle within and the bigger circles outside, these are all primary, secondary, and tertiary gene pool. What that means is, when we go right within, all these plants which are very closely connected, they are situated within. So all the plants, let's say right in the inside of the circle, they're all very, they are like all within a very same species, or probably from the same plot or everything. No, as and as they become different, they begin to diverge away from each other. So what we see is, if you see this tail emanating from center and going outside, so inside, obviously, we will have different varieties, and as we go out, slowly those the divergence, those varieties, they become wide enough, they become diverse enough to become species, and then different genera. So probably by the time you are reaching the last circle, the outer circle, they have already become different species and different genera. So in between, so this is the germplasm. This is actually how happens. Supposedly all these crops that we are eating, whether it's vegetables or the cereal crops, when when we are working on crop variety development, so obviously we need to pick genes from the best genes from anything, everything, even from closely related species and even genera. So there's a need to make a gene pool, so gene bank. So a gene bank will typically it will have something like this. So they are all interconnected. Right, now the next thing is, since we have already discussed that all the life forms, they are interconnected, then how do we draw the boundaries? That is species delineation. How do we do that? And then this question is quite important. What is the driving force behind speciation? What is it that causes speciation? Now, immediately this point will not be clear, but as we go down further into the talk, you will realize this question is quite important to address before we answer whatever where to how to draw the species boundaries. Now here, classically, what we mostly know, that is what, when we start our educational career, academic career, this is what we are taught, that species is a group of freely interbreeding individuals that can produce fertile offspring. So we can say, okay, we know a tiger, we have a tiger, we have a tigress, we have a donkey, we have everything. But then, let's say there's a tiger, and then there is a let's say a lion, or no, okay, tiger and lion, they can crossbreed and they make a liger. Okay, but the liger cannot produce children. Similarly, you have let's say in the plants, you have triticale, you have wheat, and you have probably rye. I think it's rye. Huh? You cross these two, and you have triticale, and that doesn't in this is not the best example to discuss, but the point here is that but donkey and horse is another one. We have the donkey, we have the horse. They are different species, but they can interbreed. But the children which they produce, which is the mule will not produce further children. Mule will be sterile. So in other words, this is the biological species concept. That biologically, only the individuals which can breed within themselves, they form the species. And beyond that, so this definition is what at the start of our career we are told that it's a group of really interbreeding individuals that can produce fertile offspring. Now, what is the problem with this concept? And let me tell you, I mean, I spent a lot of time going into literature, and I personally have, I've been through this period of 250 years, of, and you will be surprised to know that it took 250 years to define species, just the species, and how to define. So it's what we normally think is such. So it just to let you know that there are some tricks involved, there are some conflicts involved here. So it has taken 250 years, and it started with Darwin. We all know Darwin now. But then from 1859 to, we are talking about 2007, that is when another guy, so an American scientist by the name of D. Quiros, he wrote a very nice, I mean, a lot of people can give credit to a lot of other people, but personally, I would give credit to this guy, D. Quiros. I mean, his publications are absolutely marvelous, and he has defined species in a very nice way, which we will be discussing also. But before we go there, look at what Darwin said. We all know, the origin of species. Darwin wrote that book, and at one point, what Charles Darwin says in his book is, I was much struck how entirely vague and arbitrary is the distinction between species and varieties. In other words, what Darwin said, that it's very hard for me to tell where a variety ends and a species begins. So how can you say that two populations, and let's say talk of in terms of populations, there's a population of individuals, let's say pak choi or anything, let's say batua, huh? There's a batua, we all know, which is no, this is it's a brassica one, and there's another one, let's say um, I'm forgetting these names now. Anyway, so when we have two different populations, and they are slightly different from each other, but even varieties are different. So when do we know that they are different enough to be so different that they can be called species and we call them varieties? So the distinction is very, and this is man-made. In nature, like already we saw that chart, in nature there's no distinction really. I mean, I don't want to extend this talk too far, but really speaking, there's no difference even between plants and animals. So if we look at the, I think you understand this point, so I should not extend it further than it is really required. But talking about species and varieties, the distinction is very small. And then again, let me not go into too much. I think the point has been made. Now let's go to the different concept, just a quick one without going into too much. One is the biological species concept, which I've already said, that only the individuals which can crossbreed among themselves and produce fertile offspring thereby. But the problem is here, what about the asexually reproducing organisms? We have banana, for example. No, banana is reproducing. It could reproduce sexually also, but a lot of banana clones, they are reproducing unisexually, and they also make new types of clones, and which are totally different. So even their variability is arising, even their new species are arising. I mean, if we look at the germplasm, banana germplasm, we will look at purple banana, yellow banana, whitish, yellowish, all sorts of bananas, small, big, triangular. I mean, there's so much, and some of them they are totally different species. So from extremely different looking varieties to totally different species, even in banana, those species are there, and it does not. In a major way, it only propagates asexually. So how come it has become different species? So that means for species biological that is just one criteria, but to say that species can be formed only on the basis of biological criteria, that is not done, and it's not there are many other things which I can discuss with you, but let's concentrate more on DNA barcoding. And the point is that the original definition which we thought is the correct one, that doesn't work. Next one is the phenetic species. Now phenetic species means, phenetic is the morphology, everything that looks together is the same, is we say okay, it's the same species, and if it looks different, we say it's a different species. Okay, so that is the phenetic species concept. But there's a problem here also. There are a lot of things which look morphologically so similar, and they are totally unrelated. Let me see if there is a picture here which I can, and I'll go back to there. Here is, can we see this picture here? No, there is a picture here. One is astrophyllum, the other one is the euphorbia. Both of these plants, we can see they are totally different. They are totally different plants, and this is a process called convergent evolution. And that means even if they come from very different lineages, genetic lineages, and they are very, very different plants, and yet they look so similar. So on the basis of morphology, we cannot say that. And it's, and here this is an example of the plants, but also we all know a much better example. We know the dolphins and we know the whales. They look like fish, and initially, that is what the scientific community thought, these are fishes. But eventually, when we studied them deeper, we came to know that they're actually mammals. So they were fish. During the evolution, they came out to the land, and they went back to the sea again. So they were basically the mammals now, because they don't they produce children, they produce offspring, and they are in every sense of the word, they are really mammals. So we have the fishes and dolphins and the whales who are actually mammals and not fishes. So they look so similar. So that means, let's go back to the same earlier thing, which is the phenetic species concept, that things that the life forms which look similar, they form the same species. No, that's not done. Okay, we go to the next one, the phylogenetic species. And the phylogenetic species says, um, this is uh, this is about the relationships, the blood relationships. Um, the word philo comes from tribe, means belonging to the same tribe. So they say that when they come from the same tribe, and when they are a part of the same population, then we can say that since they're they've been moving together, they're part of the same population, and we can say that this is one species. And that again had problems, because within the same tribe also, within the same looking population also, even the genetic origin can be different. We don't know where from things are coming, and from looking from the human's point of view also, we look at the Europeans, we look at the Indians, and now a lot of DNA fingerprinting is being done, and you find out that we can all look into the YouTube and everything, and they do 23andMe. When you look at your genome, and you say, oh my god, I'm 23% African, I'm 10% Indian, and I'm only 60% European. So what it means is, even though you belong to the same tribe, you belong to the same community, you belong to the same population, where you're coming from, still that is not a guarantee. So that means even that cannot be used as a criteria, just because you happen to be moving as a population, that you can say, okay, you belong to the same species. Now let's go to another concept, and that is the monophyletic. Now monophyletic group says, this is, I don't want to go too deep into it. This basically says that even if that within, like I said, uh, sorry, this background noise. What it means is basically that lot of I give you an example of let's say a European person finding out their genetic lineage, and he finds out that his genome is 23% from Africa, 10% from India, and maybe 60% or something from Europe, and something from Russia or China. So what it means is that within a population there are different lineages which are coming together. But if we can pick up just one lineage which is genetically just pure, and we know that the ancestors are coming from one genetic lineage, and we can say that is a species. So this is what the concept of monophyletic species is. But then again, even this is not correct, because here the what they said was, how will we ever find out, because the ancestors are dead? We cannot say that this is coming from let's say there's an individual coming from that a group of individuals, or a group of individuals which are living together, existing together, is called a population. That this population is coming from a very same male and female parents, and they came from their same ancestors. I mean, the only way to find this out is, we cannot find, we will never have their DNA, we will never have their blood samples, we'll never have the leaf tissue. No, the only way to find that out is by looking at their morphology. And when we look at the morphology and we say, okay, we say humans look very similar to apes, so that means humans must have come from apes. Okay, and then you make a chart and we will discuss phylogeny, and then you look at the characters and we say, okay, apes must have come from the monkeys, and the monkeys may have come from this here, and similarly in plants also. So that means this phyletic means genetic. This genetic relationship, ancestral relationships, are also based on morphology. So we are deriving uh, genetics based on morphology. So this is really speaking not a genetic uh, basis of defining a species. This is a morphological species. So we cannot call it a truly monophyletic or phyletic or monophyletic. This I will not go into. Now the ecological spectrum. This is another concept. So even if now by this time we already know that we cannot depend on, let's say, morphological characters as the criteria, we cannot, as delineating a species. We also cannot depend on the biological species concept. We cannot say that if the individuals within a population, they can interbreed freely and produce fertile offspring, they belong to the same species. That we cannot say. So we have looked at 3 or 4 concepts, and then mind you, there's something like in one of these earlier slides, that is something 24 different concepts. But now here we talk about ecological concept. What this ecological concept says is, if now, for example, in this case, what it says is, the populations or the individuals which share the same ecological niche, they will be called the same species. So this is called the ecological species concept. In this case, again, we've already discussed. For example, we have 2 very different looking plants, Euphorbia obesa, and we have Astrophyllum asterias, and they look very similar, and they exist in the same niche, but still they are very different. So what is coming in the same ecological niche doesn't have to have to be the same species. And we talked about the fishes also, just because and that basically means that even ecology, and there are some good examples which I can quote, but I will not go into that, where lot of things, this ecological species concept makes sense, and when other concepts cannot explain how individuals or populations can be delineated from other species, ecological concept can be quite handy. But again, this cannot be used in a wholesome way. Like here it is that a lot of life forms coexist in the same or similar ecological niche. So even then we cannot say this is, there are other reasons also why ecology cannot be the criteria, but I will not go into that. So now, so far what we have tried to say is that, how do we define species? How we say this is a banana? Because banana will merge into another crop, which is very much like banana, and that will merge into another, into another, into another. So the thing is, it's since it's a continuous chain, it's a continuous tree of life. One merges into another. Where do we draw the boundaries? That becomes very difficult. And all of these things which we have discussed, they are not able to delineate the boundaries of species here. So then the question came was that, is there something called a unified concept of species? Can somebody, you know, define what a species is and how to delineate species? And this is where De Quiros gave a very nice, proposal. He said basically, so I will not go into the text here. What he said, he defined a species as simply, very simple definition, a separately evolving metapopulation lineage. Now, let me not confuse you with too many words. What this basically means is, when a population which is existing together starts diverging, this becomes slowly, it takes a different identity and it becomes different. Okay, and so this could start with a genetic drift. This could, and speciation is happening all the time. So slowly what happens is, from a large pool of individuals, give me, I mean, there's a very nice, nice picture from, especially by the same guy, De Quiros, and I would really like to show you that picture. Let me see if I can. That paper I have already sent to you, but from the same paper, let me try to show you that picture. Just one second. Give me 1 second. Yeah. So if you could, I hope my screen is visible. In that case, this is a very nice, forking picture. If we look at the bottom part of the picture, we see there is a population here. So this fork, we go to the common part, the stem. If we go to the stem, this is a species where it says one species. Now this species slowly, due to some reason, this could be genetic drift, this could be change of climate, or there could be, like, there's a partition, somebody has built a bridge in between, so the people on one side, one plants growing on one side, the other one on another side, or there's a lake which has come up, or somebody has constructed a dam or something. Anything can happen, and because of which adaptation is happening, evolution is happening, and slowly different characters are emerging. Now in this case, it starts from the stem, and slowly we see, the colors are, so one, the same population is diverging in two different directions towards the top. And a time comes when one population becomes black, the other one is white. Those differences are so stark that they become totally different species. So this is how speciation is happening. Now we go back to the same point which De Quiros said. So what he said was a separately evolving metapopulation lineage. When a population which is working together, moving together, and so which is evolving together, metapopulation, that is called a species. That is a species. Okay, they say meta population. Meta basically means that it's not a very homogeneous thing. There are from one individual to another individual, there are always some changes. I mean, I will not be totally similar to my brother or to my sister. So all these things, it's not a very population. In this case, my case, it's a population. But when we look at a bigger population, species, at the level of that, we are talking about many populations coming together. It seems somebody has joined. Let me let them in. So what De Quiro said was that the moment a group of individuals, they start moving together, they start evolving together, and that's a species, that's an entirely different species, and slowly that drift is happening. Now the second, so this is the process of speciation. And how do we define species? What he said was, to separate species, anything can be used. We can use the ecological species concept, we can use the biological species concept, we can use the phylogenetic species. So all of these are criteria. So if something is ecologically different, we say, okay, it's a different species, and we can delineate based on ecology. If something is biologically different, it cannot sexually reproduce together, it cannot cross with each other, we say these are two different species. If something is, let's say, they have a genetically, they're coming from genetically different sources, different populations, different ancestry, we can say, okay, this is polyphyletic, this is not monophyletic, and they fall under the category of separately evolving metapopulation lineage. Speciation has already started, and they are going to, and the moment it becomes big enough, the moment it becomes visible enough, we'll say, okay, these are delineated. So this should explain the concept of species. Now, one thing is very clear here. Now let us look at, because what DNA barcoding does is, basically, this will tell us a plant belongs to this particular species. Now, let us find out what DNA bar, and we will go back to DNA barcoding now, and we'll see what DNA barcoding can do or it cannot do. Now, what is the role of DNA barcoding? I'll go back to the initial slides where we discussed what DNA barcoding does. Like I showed in my own case, I had the pak choi plants samples from which I took the DNA, I did the sequencing, and those sequence was matched against a database. And the database tells, okay, you took the DNA from a plant and you are trying to compare your sequence with the database sequence, and our database tells you that there are some specimens, there are some entries here which match totally with the sequence that you are giving, and these two sequences match. So that means your plant is, let's say, pak choi, or it's Brassica rapa chinensis. Okay, so what DNA barcoding here is doing is, they are simply they're simply comparing the sequence of our plant or our microorganism or our animal with the sequences which have been submitted, which have been deposited into a database. Now the point is, somebody who deposited in the database, if they are not able to identify in the first place, they cannot tell that, okay, I'm putting up the DNA of pak choi. If they cannot tell, I cannot. So somebody has to do it first. Now, who does it first? Who does the identification? Now, that is an identification which is done by the taxonomist. So the numerical taxonomist who actually look at the morphology. So the identification, the species identification, is basically the job of the taxonomist, the job of the botanical, taxonomical, classical taxonomists. They are the people who tell what are the characters. So they look at the characters. So based on the character, those things are defined. And after that, once a species has been defined, those samples, many people begin to identify them, and that DNA is put into the database, and they say, okay, this sequence is coming from this species. And if somebody finds this species, you don't have to look at the plant. You don't have to identify. You don't have to be an expert. You don't have to be an expert botanist. You don't have to be an expert taxonomist to identify that this species is, what is this species. Simply from that segment of DNA, from the database, we can tell you this is this species, and that is what the DNA barcoding will do. Right. So any questions, please, keep stopping me there, and we can discuss right there. But I'll move on. What are DNA barcodes? So in the first slide, what I said was that DNA, these are small segments of DNA from which and those sequences can be used to discriminate life forms. What are those barcodes, or what those small fragments of DNA are called DNA barcodes. When this was started by Paul Herbert, they only had two original loci. It was, these are called RBCL, METK. These are all different genes, like RBCL, this is a ribulose gene, ribulose nuclease gene, something. It's an enzyme. METK is a maturase K, coming from this maturase K gene. Similarly, then ITS is inter transgenic sequencer spacer, something like that. So they are coming either from the genes or they could be from a non-genic area also, but these are sequences. Now what these will do is, so we simply, now here are these, let's say in this case, and there are 5 barcodes. In other words, there are 5 small segments. These are called RBCL, METK, TRNH, PSBA, and this is between 2 genes, TRNH and PSB genes in between area. So intertransgenic sequence, ITS. And no, this is the sequence. This is a bit different. So these are 5 different small segments, and if we can look at these sequences and compare them with the database, we can identify the plant and we can tell which species it is coming from. How do we do this? Here is the workflow. And the workflow is, we obviously we move from left to right. First, we take the sample. This is called specimen collection. Then vouchering. Vouchering is, vouchering basically means we got to note down all the aspects. And that's a good point to discuss here also, because we need to note down the location. We need to note down the climate. We also need to note down probably what are the other species which are found around this, because even that helps in identification. So vouchering basically means when was all the details, time, date, place, all those details, they have been noted. And we will come to this point that DNA barcoding alone cannot always identify. This helps us in identification, but many times we need to do a secondary criteria to sort of narrow down our identification, and to be whenever there's a conflict between 2 or 3 different identifications, we are not sure, and this vouchering can help. Okay, this plant came from this habitat and this cannot be this, so this information can be quite handy. So we need to do the vouchering. Then obviously somebody is there, let me get them in. So, and then we extract the DNA and then it's sequenced. Once the sequence is available, we can match it with the existing database. This is bioinformatics analysis. Then we blast it with the existing database. That's the analytical bit. And then we feed it. This is called blasting. Then we go into the database, and I'll show you the slides. And then it will tell us, okay, what plant it is. This is not the end of the story. This is one part, and we'll see how it, the other thing. But this is the basic process of DNA barcoding. Again, I think this is something which is pretty basic. We have discussed yesterday also. So what we will start is, once we have collected the samples, plant samples, we will extract the DNA, and from DNA we will do the PCR using, just one second, somebody is there. So we do the PCR, and once the PCR is done, using those primers against those genes or those sequences which have been defined, like METK or RBCL or ITS, for using those primers, we do the PCR, and then these bands are cut out. So these bands can be cut out, and this DNA is then purified, and then this is sequenced. So we can say these are bands of different types coming up, and this DNA is cut out and purified, and this is sequenced. Then once the sequence is there, we can blast it to a database like NCBI. I've given the link here, and you get a variety of identification. So you can see these are called hits. There are a lot of hits, and they will even tell you what is the percentage match. So this were based on percentage match, how much of the sequence is matching. This can be 99%, this can be 100% match, this can be 85%, 90%. So based on that, a category, we have a top to bottom, it's a hit list which is formed, which says, okay, the best identification, 100% identification comes from a species called, okay, whatever. In this particular case, if you can see, it says, if they are doing some fungal antidote, they use some name. Okay, so that is how and again, in this case, there is a, the species are something like menziaceae. The top 10 hits are menziaceae. The next is cubensis, and then, so in this particular case, we have 2 or 3 types of different identification, but the top is menziaceae, and then it gives something like cubensis or something. So these are some fungal so these are some fungi, and so we can say that the best hit is mensite, and so most likely our species is going to be, our plant is, or the microorganism is going to be this. Okay, this we have discussed. Okay, now there's another point here. Since by this time we already know what DNA barcoding means. Okay, now we shall be discussing about what it can do, some of the things which about limitations and other things which are required in addition to DNA barcoding in order to do the plant identification. But before I move into the other bit, and that also includes phylogeny, we'll need to do, we need to do phylogenetic analysis. But before we go into those steps, the basic protocol, the basic process of DNA barcoding has already been discussed. If you have questions, please feel free. So let's take a pause here and let me wait for any questions if you have any queries. If not, then I'll move on. So it seems there is no query. You can also put it into the chat in case you have, or but I don't see there is any query, so probably I shall continue to the next one. Okay, like I said, all of life forms, they are interconnected, and that means delineating, separating one species from another species becomes very difficult. And that is the reason, if we have there's something called barcode gaps. Now, yesterday we talked about dendrograms. Like I said, if you remember that what DNA, the dendrogram, this is a dendrogram here. What dendrogram does is, when we look at the genetic matrix, when we look at the DNA matrix, it's only comparing 2 species, 2 individuals together. But if we want to look many individuals together, then we have to look at a dendrogram, because that is when the interrelationships, they become closer, they become more visible. Here, if you see, if we go, let's say, to the lower level. Okay, let's start from left to right. From left to right, it's very clear. There are 2 major groups here. There's one smaller group on the lower side where there are only 2 species. And there's another bigger group on the upper side where there's something like, I don't know, maybe 13, 15 of, something like 13 to 15 species. Okay, now it's 14 other species, or maybe 14, 17. Okay, so let's say there are 12 to 14 species on the other side. So these are 2 different groups. On the bigger group, if we go further down, that can be divided into 2 smaller groups. The groups, there's one, the lower group carrying something like 5 species, the upper group carrying something like 9 species. So there's another 5 and 19. So again, from the other group, so you can see that the species which are very close to each other, they are placed close to each other. They are not far away from each other. They are in a smaller group, and the bigger ones, they are far apart from each other. Now, what is the DNA barcode? Now we know that the lower two species are very different from other two species. So obviously this is in this case, so this information is coming from the barcodes, from the sequences. The sequences have been converted into a dendrogram. So if you remember yesterday, we discussed how we can convert a matrix, a distance matrix, into a dendrogram. So basically, the using the same data. So just the way we were using molecular markers, how many? I think I should go back to the same before you. If this slide makes sense to you, let me for a second go back to the same slide, and then you will it's the same thing we are. So in the earlier case, yeah, what we discussed yesterday, we were talking about the information was about similarity or difference was coming from different markers. In this case, it's coming from nucleotides, from different sequences. So if I go back to the same slide here, let us say oops, sorry. Okay, I think that will be so if we look at this picture, which we discussed yesterday, so we know how the marker data is being converted into, into 0 and 1 score, and the marker score, it's converted into a score, and then this is converted into the distance matrix, and from distance matrix we can convert it into a dendrogram. So with the distance matrix, we can only compare pairs, like number 2 variety can be compared to number 3, or number 2 can be compared to number 5. So here only pairs can be compared. But when all of this, so we can use statistics, and we will be doing this, and we will, I got a call from ma'am, Dr. Manjunath, and I think she's very keen that the students discuss phylogenetics, and probably will be doing the dendrogram making. So we'll come to this point and it will be even more clear. But this data can be converted, and so all of these relationships can be used. So the only difference is here we are using different markers, like the lower one, upper one from the same variety. In case of sequences, so we can do this. Here is the sequence. Probably we have one slide which is shows information on the sequences also, and we had a look at that data. This is the one. So we discussed this, the person 1, person 2, person 3. Okay, so this is how DNA barcoding will be also. In this case, what we say is, if you remember, we talked about number 6 and number 7. If you see the A here, T here, A here, A here, A here, and T here. So again, hello. Yeah, go on. It's not visible. Yeah, yeah. Uh, okay, but the screen is visible. Yeah, no problem. So I'll remove this, and then I'll come back to the same earlier slide, earlier presentation. Yeah. Thanks for letting me know. Yeah, okay. So this was the earlier one, and I'll go back to the presentation, the earlier presentation, just to let you know that how the sequence is. So you don't have to. It's not just the bands based on the bands only. The fragments you can make the dendrogram. Once the sequence is there, again that can be converted into. So I hope the presentation is now visible. Yeah. So again, coming back to the same, barcode gap. So if you can, so this is what is called the barcode gaps. These big things, and they make it easier to delineate different species. So if the two species they do not show variations, if the two and individuals they do not show variations in the DNA barcodes, we shall not be able to know they are from the different species. So from this slide, what the take home message will be, in order to identify a species, it's very important that there are barcode gaps. If they merge into one another, like in this case, let's say there are, if you look at the lower level species here, in the lower down in the hierarchy, there's one in the middle, like there's some phylla and anisophila, and there's one linearophylla, and there's one ciliata. Both of these, all three of them, they are so close to each other, and we cannot say they are different. Even if they are very different, but we don't get the data where there's a barcode gap. That gap has to be there, and that is so barcode will help us identify the species. And the other thing which had to be said here is the role of phylogeny. So you will realize that one is the barcode hits. So when we are doing DNA fingerprinting, one, like we already discussed, the hits, the blast hits. So we do the hits and we say, okay, there's 100% or 99% match. This is the best choice. If you remember, we talked about those fungi there. Yeah? And the second will be making the dendrograms. Dendrograms will tell us where they are close, and they can show us the relationship. So what is close to each other, which can form a different species, which are the individuals population which fall in the different groups. So they can also help in species. So if one individual of our choice is close to an identified species, we can say, okay, our individual is close to that. And if there's a conflict between and that can be used to resolve conflicts. So the point I'm trying to say here is that DNA barcoding will be used. So DNA hits will be, blast hits is one criteria, and phylogeny, making dendrograms, that is another criteria. No, I'm talking about examples of the DNA barcoding. This is DNA barcoding. The beauty of DNA barcoding is, you see, let's say medicinal plants. In this case, there's many of us might know this. There's a medicinal plant called combretum, and if we look at the fruit of this plant, they are all so similar. In this case, these are 7 different species, but we can clearly see they all look so similar. Trust me, the morphological taxonomists, if they go to the field, they go to the forest, it will be very, very hard to find out. Even when the tree is there, when you are plucking it, it will be very hard to identify that this is the same species. And I don't know if you guys are following some of the news. A few days back, there was a news from Thailand. Somebody picked up the wrong fungus, and they died. Somebody died. No, forget about fungus. Let's talk about. We all know aloe vera. No, aloe vera. There was an interview interviewed in some European channel, and this lady was telling the benefits of aloe vera. And she was probably a dietitian, and she was telling people how to eat this, and all the good things about aloe vera. And what she did was she took up a sample, and this is a sample of aloe vera. She cut it down and she got the gel out of it and say, so that is the way to eat it. And to everybody's astonishment, the moment she had it, a couple of minutes later, she began to show very abnormal behavior, and eventually that the interviewee, the lady who was giving the interview, who was telling everybody, she was a dietitian, and she's telling people why to eat aloe vera, how to eat aloe vera. She died. Can you believe it? An expert giving talks on aloe vera. She took a plant which looked like aloe vera but wasn't an aloe vera. She died. And we, all of us, we know there are times when, let's say, somebody takes a cucumber, it's bitter. They take cucumber, it's not supposed to be eaten, they die. So what I mean to say is that, certain things are unavoidable. I mean, we all eat cucumber, and the only thing we can say is, okay, don't take the bitter one. That's fine. But sometimes, and in that case, so what I mean to say is that, medicinal plants and even edible plants, identifying them and the way they are being sold in the market and the way they are being presented as medicinal plants, especially with the post-corona period, COVID period, Ayurveda has really picked up, and everybody is talking green. So, and that is also the reason why we've become a bit more wanted. Agri-biotech is really wanted. In that case, all these identifications are very important. And if you look at that, a lot of people are doing identification, and why not? One of you, there's no reason why you can not set up your own company and start looking at the Ayurvedic samples, and you start, and that's business. And those are the sort of things I'm sure you guys should be looking at once you are done with your course. You don't have to look for jobs. You start creating jobs. You start making your own businesses, and that this is really the way to go forward. But coming back to the point, the uses of these plants these days, the potential of DNA barcoding in identifying identifying the adulterants within the Ayurvedic drugs. And mind you, what we get in the market is, we'll get a part of stem, we'll get a part of ground powder of a leaf, we will get, let's say, a fruit. When the fruit looks so similar, you cannot, I mean, because the plant is not there, even the traditional botanists, taxonomists cannot do anything. You need the DNA. So you extract the DNA, you look at the DNA, you do DNA barcoding, and that's it. So for all the drug inspectors, you see, even the drug inspectors, I'm not sure, because in India, your own university or your own college is an example. How many people are being teaching all these advanced? They're not very advanced. I mean, the world has already reached there. So how many people are really learning and teaching these things? Not many. So, you don't expect even the drug inspector would be doing all these things, but this is something which has to be picked up by society. This is something which has to be picked up by by our social system and the government system also. And ideally, they should have the facility to do DNA barcoding also. And those are all your potential jobs, not just in private sector, but also in industry, in academia, and also in government institutions like drug inspectors. Again, this is another example of a plant. Again, it's again combating species. But if we look at the inflorescence, the top two, can we tell these flowers are different? Nobody can tell. Similarly, if we go to the bottom three, they look so similar. If we look, if somebody gives us the flowers, if somebody gives us these inflorescences, probably even the best of taxonomists will not be, they can make mistakes. Just the way that dietitian giving talks on diet, giving talks on aloe vera, right in full view of public, right? I mean, this is totally shocking. So that shows the reach of DNA barcoding and how far this can really be of use to society. Before I go any further, I think I should. I'm not sure if I've included enough the slides. Uh, I'm not sure if I've included some more slides, but I should say here that DNA barcoding, like I said, the success of DNA barcoding is not total. I mean, in plants, it's been found that 70% of the cases can be identified by DNA barcoding. In case of animals, 90% of the animals can be identified by DNA barcoding, rough figures. And in case of microorganisms like bacteria, fungi, they're almost 100%, 99, 100%. So far, and that is where it is most important. DNA barcoding In fact, one of my friends from she is happens to be an assistant professor in one of the universities from Chandigarh University, and she wanted to identify, and I gave a talk. I was invited for a talk to their university, and she's doing a PhD, and she couldn't. She was finding it very difficult to identify her species, and she couldn't identify the bacteria. And the thing is, I mean, for plants, you can look at the floral structures, you can look at leaves and all those things. What do you look at for the bacteria, for the fungi? It's very difficult. There, DNA barcoding is all the more successful, and the success rate is anywhere between 95 to 100%. So DNA barcoding is even more successful in the microorganisms and in animals. And of course, in plants, the success is not total, but still, and they are continuously these technologies, they are evolving continuously. And let me tell you, I mean, about a week back, I had a discussion with some of my earlier colleagues from John Center in UK, and we had discussion of some of the plants which they are trying to identify, and they were trying to resolve a species conflict, because there are certain plants which form different species, but they look though similar, and they didn't know how to do it. And like in this case, we use only 5 barcodes. If you remember, I talked about MATK and some other things. Um, if we go for higher level identifications, these 5 are not enough. So when I say 70% of the plants can be identified clearly, if we can improve these technologies, if we can increase the number of markers from 5 to 10 to 20, and in certain cases even up to 1,000, then we can resolve even the tricky cases, and that's being done. In fact, like I said, about a week back, I attended this talk from John Center, and they resolved a very tricky case in one of the families, and they could tell them apart using something like 1,000 DNA, 1,000 markers sequences. So in that case, when we are talking about 5 barcodes, they will give you about 70% of the plant species, or they will tell you. But if the other ones, they are not found out, there's no reason to panic. I mean, if you really want to do, if you want to go into deeper research, this is a very open area of research. You can do it, and those things can be done. And obviously, that's also the way. If you really know the thing, if you are into the trade, and if you go into industry, and you can solve the tricky cases. So what I mean to say is that this is very much an active area of research, and it's also open to you all for research. If you really, if you like to be Google, if you like to be into green research, and if you feel interested, this is also an upcoming area. This is also very much a hot area which can be taken up, and best of institutes across the globe, they are working on it. Now, some of the remarks I would like to make on DNA barcoding is, when we are doing DNA barcoding, if you remember, okay, so this is the slide. In fact, this is what I wanted to show you. If you look at, on the left-hand side, this is animals. On the middle, this is plant groups. And in the right-hand side, this is microorganisms, right? Like I said, so this is another thing. The barcodes can be different for different. So in plant groups, in the middle, B group, for example, we have ITS2, ITS, MATK, PSBA, TRNH, and RBCL. So for animals, these things will also work. They also work in animals, but not to that extent, not to that accuracy. There, this is one CO1. So it's something like carboxylase oxidase 1 gene. Similar, then 16S RNA is used, cytochrome B is used, ITS2 is used, same as in plants, and then 28S rRNA. So the barcodes are different, but then ITS is present in both plants and animals. So the barcodes become different. Similarly, in microorganism, we say they have COI. COI is the CO1 is the same as in case of let's say animal groups. Similarly, 16S RNA, RPB2. So the barcodes can be slightly different across different groups of individuals when we talk about animals, plants, or microorganisms, and also the success rate varies. Another figure I would like to discuss with you is, you see, there's a difference between molecular markers and DNA barcoding. When we are doing DNA barcoding, this has to be quite exact. because we are doing identification based on a sequence, and that sequence has to be exact. I'm not sure if many of you are aware that the Tag polymerase that we use, this carries errors. When we are doing DNA barcoding, those errors cannot be allowed. Just one second. So once we are doing DNA barcoding, those differences, those errors in the sequence, they are not acceptable. So that is the reason we use high quality Tag polymerase. In this case, you can see there's a different gradation. A lot of people use Tag enzyme, Tag polymerase. That is not a good idea in case of DNA barcoding. There are different other, like they have Kaizen Hotstar, they have Kappa, and then they have Platinum Superfi, and then they have NEB. So this is the one I use, that New England Biolaboratories, NEB. And this was high, this was quite good. The number 2, this was quite good. But again, there's another one, Platinum Superfi. So the whole idea is you have to be a bit careful about the enzyme that you use. The Tag polymerase or the polymerase enzyme that you use has to be higher quality, because we want the sequence data which is reliable, and there's no room for making errors. Now we come to the limitation of DNA barcoding, what it can do. And so, like along the presentation, we have been discussing that there are certain things which DNA barcoding cannot be do. So among plants, recently diverged species, polyploids. Why I say polyploids? Because you see, in polyploids there are multiple genomes. Polyploid by definition means, let's say, in tomato is a diploid. So there are two sets of genomes, haplomes, one coming from male, one coming from female, and you have the diploid, right? In case of wheat is hexaploid. So in other words, there are a set of 6 genes for every 6 alleles for every gene. Even if one carries a different mutation under the background of 6 different, that data will not be visible. So when there are multiple genes and multiple alleles of the same type available within the same plant, then analysis can become tricky. It becomes difficult to find out, is it T? So for polyploids, they can be a bit tricky to deal. Similarly, if there's nobody has done DNA barcoding, nobody has done sequence those DNA bits and deposited that database, information on the database, then we will not be able to compare with a known sample, with a known plant. So if there's no match in the database, we cannot do identification. And so and that is why people all the time, they are giving entries. Everybody wants to be famous, you see, and why not? This probably there are certain plants which are growing only in, let's say, Sholapur. Nobody in other part of the world would be concerned. So some pandemic species, some plants which are which are really used in this in isolated areas, maybe they don't have any match. And really speaking, this that gives us a feeling of deja vu. Let's connect this with corona also. When corona, for COVID also, when vaccines are being made and when these identifications are being done, if that strain is not, supposedly in India, in one part of India, a strain develops which is very different from others, the vaccines may not work there. So the vaccines, when they make vaccines, they will be looking at the sequences which they have analyzed. So it's a good thing that we have Indian companies who look at the sequences of Indian variants, and then they make the vaccine. But if that information is not there, that's it, you get left out. Now let's talk about the DNA barcoding also. If the information in the database is not there about a certain species, we won't get any match. So, and that is, again, for you guys, there is a chance to do discoveries, to look for the Indian germplasm, to look for what is happening around. So getting left out is not a good idea. So what I mean to say is, when we say no match, that means there's a possibility of always writing research papers, and there's always possibilities of doing higher studies, PhDs, and there's always a possibility of being famous, even if you don't do the, you give an entry. I mean, my basically my aim is, if I get students in the lab, in our labs, I intend to put these things in databases. And you are, the student who comes for training, well, you feed the name in the Google and you say, okay, this student found this plant and this sequence was given. You are rich. I mean, at least, that adds to your CV, that look, this is the sequence I found out and I submitted into the database. What I mean to say is that there is a possibility of doing research and also trainings here, and you could identify species, or if not, nothing else, your own sample. The minimum you can do is submit your sequence into the database and get your name printed there. Okay, well, you are noticed. Then another thing is poor quality sequence. If somebody is, obviously, you give the sequence and you haven't done the identification, you are not very sure that your plant is, let's say, Mangifera indica, or your plant is whatever, Borhavia this. You give the wrong identification, then other people will also match with that and they will also identify it wrongly. So poor quality sequence information, if you've used the wrong enzyme and then done the identification, so other people in society will depend on the information provided by a lot of other people, and that is how hits are formed, and that is why we depend upon 100% or 99% hits. And that is when that gives us more confidence that, look, everybody identified this individual, this sequence as this plant or this individual, so we can say with confidence that this again, multiple matches, like I showed you in the example that we just studied. There were 2 or 3 different hits, and we go for the best match. Now, traffic light system. Okay, what time is it now? It's 3:20 already. Um, could Before I go to this bit, could we take maybe 5 minutes break? It's been an hour and 20 minutes, so we take 5 minutes and then we get back into it. Okay then. Okay, okay, please. Shall we start? Shall we start? Right. Is the screen visible? Okay. No. So this traffic light system, like we discussed in some of the earlier slides, that especially in plants, using DNA barcoding, this is not a foolproof. You don't always get the identification. So broadly speaking, it's about 70% identification. In certain taxa, like in certain genera, in certain families, DNA barcoding is highly successful. And in certain areas, this is not that successful. It's not very, it doesn't give very good results. It's not very successful. And I discussed some of the cases there, like the recent evolution, recent divergence or reticulate evolution. There are certain reasons why this happens, but I guess I should not discuss into those issues, because for the beginners, I don't see much of a point going into those areas, which could be a little bit of a put off. But broadly, we can define different species on the basis of a system called a traffic light system. For example, green, yellow, and red light. So the species from the families, from the genera, where identifications are highly successful, and which is very amenable to DNA barcoding, they are given like a green light. So if the species of our the plant we want to identify, and we know roughly where it falls, no, this is a bit tricky. I mean, we don't know something and we already are making guesses. That So that is why, it makes sense to make guesses sometimes, because if our guess is that, look, our plant belongs to this particular family, and we already know that that family is giving a tricky result to DNA barcoding, based on how difficult the DNA barcoding, or how amenable this is, the family or that genus is to DNA barcoding. This light system is used: green light, amber light, and the red light. And this is, for example, orchids. Like they say that in case of orchids, it's yellow light. So again, when we do DNA barcoding, just to let you know that we can be forewarned, rather than trying too hard. So that also shows us when to use alternative methodologies, or when to increase our, the usage of our markers. So probably we can use more number of DNA barcodes to do the identification. So this traffic light system can be handy in case of, if we already are aware about what roughly where our plant could fall. Now, we partially discussed this concept of integrated taxonomy, and what that means is basically, when we want to do identification, simply doing DNA barcoding may not be enough. There are times when there are conflicts. Like, we look at the hits from the sequence hits, we get 2 or 3 different species. There's one hit, there's a second hit, there's a third hit, and then if they are close hits, it makes sense that in addition to the molecular results, we can look at the morphological result. And this is how even I did my identification. In my case, I was doing, in my work in Malaysia, I was doing identifications between subfamilies of pak choy. So pak choy is Brassica rapa. Rapa is the species, but within rapa, there's Brassica rapa pekinensis and Brassica rapa chinensis. Now, in order to, just one second. So molecular data, there are times when molecular data may give you 2 or 3 different choices. When you start comparing the morphology also, for example, with the molecular data, you get 3 species. And one of the species, 2 of them are widely diverse, and you can look at the morphology. And then you say, oh, this is not even possible. So there are certain area where morphology, where molecular data shows they look very similar, but morphology tells you clearly, oh no, this is very difficult. So when there are conflicts in molecular results, you look at the biomorphological data, and you could solve those conflicts very easily. And the other way also, for example, like I showed you in that ecology species conflict, that there was one euphorbia species which looked very similar morphologically to another species like cactus, but when we looked at the molecular data, and they are totally different. Similarly, biochemical and ecological and genealogical, means genealogy is family history. So all these information, they can be combined, and what could be tricky according to one parameter might be very easy to to figure out based on another parameter. So there's a need for integrative taxonomy, and that is the reason you see. I mean, if you look at it this way, that also means the taxonomy in today's, whether it's not about gene banks and in the industry, but also when we are looking about, let's say, Botanical Survey of India. In fact, 2 of my students, I gave my course on DNA barcoding. 2 of my students students. One was from HP University, Solon, and the other one was a senior scientist from Botanical Survey of India. And of course, I mean, a lot of my students are very senior people, and we are good friends now. But the thing is, they were having problems in species discrimination. And DNA barcoding is extremely useful. And in fact, I have taxonomist friends, botanist friends, senior friends, who have problems with identifications using molecular, morphological thing. And to my dismay, I mean, the state of affairs in India is so bad. I mean, when I talk to my friends in universities, many of them, they are still using that outdated, that Bentham and Heck, Bentham and what is it? System of class plant taxonomy classification. So which was made in 1880 to 1806. I mean, we are doing identifications from those, and trust me, if you look at your own university, go to the Department of Botany and you say, which system are you teaching? They will say Bentham and Hooker. Bentham and Hooker went out of the window. People across the globe, nobody. You go to the Kew, the botanical gardens. You go at. They are all talking about APG 3, APG 4, definitely APG 4, if not 5, no, but I think APG 4 it is already there. And they are using DNA barcoding, they're using molecular data. And that is also the reason many times our people, they write papers, they use a name of a plant, and that already the name of the species has been changed. For example, I remember this babul plant in my, when we were, in our early days, this was called Acacia nilotica. And if we look at the same plant now, it's not acacia. This is by a different name. I'm forgetting, but it's a different name now altogether. So all the genera and all these species, when we talk about taxonomy also, the name nomenclature and the species ascriptions are changing drastically, and that is where the molecular data is required. And that is the reason why the molecular scientists, the molecular researchers, they are required in Botanical Survey of India. They are also required in the universities. And in fact, our system is so outdated. And in fact, I've been even talking to a lot of the people, people like obviously a lot of HODs and higher levels, but they are it's a very dead system. It's like they say, okay, we just do what the UGC tells us. UGC says, okay, the Bentham and Hooker system, and we keep doing it like idiots. You see, I'm sorry to use that word. That's the exact word to use for these people. They won't question UGC. No, UGC is doing something from 1882, and they will be doing okay Bentham and Hooker. Nobody knows about APG. And I've got at least 10 senior people who are professors in the universities, and they're not willing to take a stand. So what I mean to say is that you guys, especially the young blood, here is a possibility. Pick up these tools, and they are required not just in identify in the drug inspectors for checking the adulterants in Ayurvedic drugs, but they're also used in our Botanical Survey of India and the outdated syllabi within our universities. And so it's very important to be updated. I'm sorry, that was a bit off the side, but it's important to say these things, because the older people, they're not willing to. I mean, a teacher, I've seen a lot of people, they won't do anything. All they do is just go to class, teach what is already there without ever questioning what they're teaching is up to date or not. Anyway, coming back to the applications of DNA, democratization of specimen identification. You see, in our times, let me tell you, I have, I did a double master's in botany, and then I did a double master's in biotechnology. So double master's and double MPhil in biotechnology abroad, UK. So in our times, I mean, I did MSc botany first, MPhil botany first before I went into biotechnology and molecular biology, and then at the end of MSc and MPhil, you go into the field, you go into the let's say garden, you go into the farm side, you cannot tell the species. Nobody will tell you. I mean, we were looking at bryophytes, we were looking at angiosperms, we were looking at a lot of other plants. You go to the garden and somebody asks you, okay, you're a botanist, can you tell me this plant? The whole country is producing botanists who cannot even identify the plants, because identification is hard. You try looking at all the, you see, if you study botany, if you look at the terminologies, even to describe a flower, you got to look at terms, maybe 100 different terms for corolla, for anther, for everything, different types of being. And so by the time you become a taxonomist, how many taxonomists are there in India? On the other hand, if you use the molecular techniques, you don't have to do anything. You sit into your lab, do the DNA stuff, do the sequencing, do the blasting, and without being doing too much of morphological identification, going to the forest and doing 4 years of botanical courses, you do it straight away in a couple of days, say 6 7 days or whatever. So what I mean to say, this is the democratization. Now everybody can be a taxonomist. Earlier, even the students of botany did not know anything when they went out. And now the time is every molecular biologist can tell you which plant it is. So this is a true democratization. Secondly, identifying cryptic species. Earlier, I mean, the professors, and I remember one of my teachers, Dr. Bambi, and he spent all his life telling us that he identified a species. In today's world, identifying species is a piece of cake. All you got to do is, I don't say absolutely a piece of cake, but it's not that difficult. What for the taxonomists, for the morphological taxonomists, this was a difficult thing to do, and they have to study a lot. These days, all you got to do is collect plant samples and start looking them into DNA bar. A lot of people have published papers. A lot of people have discovered new species based on DNA barcoding, and why not? So that's one area you could take up. And also the cryptic species. Cryptic means hidden. A lot of species which look so much like the other one, so much like I told you about that lady who died, that dietitian who died because of that. She couldn't identify aloe vera. They look so similar, cryptic. They look very similar, but they are different. How to tell things which are looking very similar, because you got to do DNA barcoding of a lot of samples, and only then you can tell that at the molecular level, at the DNA level, the differences are there. And then you have to find out other ways. So you can identify cryptic hidden species, then new species discovery, taxonomic refinement. Like I told you, I mean, just 7 days back, I had this talk with my colleagues in UK, and they were not able to find out, they were not able to distinguish between two tricky species, and they used not 5 but 1,000 markers, 1,000 DNA barcodes, and they were able to clearly discriminate between those two close species. So where morphology fails, molecular taxonomy can really help. So this can do refinement, and that is how, by in today's APG4 system of plant taxonomy and systematic taxonomy, has really been able to come up from the old outdated things like Bentham and Hooker of 1882. So taxonomic refinement, much of it comes from molecular data. All the modern systems of taxonomy and systematics, they are based on molecular studies in addition to morphological studies. Check misidentifications, add mixtures. That's just the same what we discussed when we were discussing the molecular markers, and when we were discussing the DNA fingerprinting. Lookalikes in medicinal plants. Oh, trust me, you go to the market, you buy Ayurvedic drugs, you get only 30% of the things which are what you really want. You start taking samples, and trust me, since I'm a botanist also, in addition to being a biotechnologist, and I also I keep discussing with a lot of roadside and people who are selling either Ayurvedic drugs or in these small tents and everywhere. They're all lost. They need help. They're not able to, even the specialist people who are selling Ayurvedic drugs, they don't know how to do it. So they get cheated, or they are not able to tell exactly how to tell lookalikes from each other. And this is where the DNA barcoding can be of big help. So please, the young blood, the young people, develop some passion, and these are the areas you can take up. Microorganism, like I said, morphologically, come on, bacteria, fungi, bacteria, it's just a blob, just a tiny dot that you see in the microplate. If it's all a white and white, all you see is white and white and white. Like this friend of mine who is an assistant professor in this Chandigarh University, and she was discussing in fact about her PhD thesis. What you see is nothing, and she had some morphological criteria, but eventually she did the identification using DNA barcoding. she sequenced the 16 sRNA, and that is how she identified her bacterium. And similarly, so I think so DNA barcoding has a lot of applications in identification of microorganisms. So if you want to take up microbiology, you cannot this will come in very handy. Plant identification from isolated plants like bark and fruit, we have already discussed. This you cannot do with the normal plant taxonomy. No, traditional taxonomy may not help you once you have just the bark before you, the fruit before you. And similarly, identification of fungi. In case of fungi, DNA barcoding is extremely capable, almost 100%, 99-100%. It will give you the identification totally correct. I mean, really reliable. Okay, the role of DNA barcoding, we have discussed that DNA barcoding, not to, it's a very important, okay, now this is a point which I would like to discuss with all of you guys. I mean, when we are doing the biotechnology, when we are doing molecular studies, the world has not caught up. The point is sometimes you move ahead of the time. Okay, it's like saying you go to America, you spend 10 years there. When you go back to the village, you will become mad because people will not be able to relate to you. You know, there's something is right and that has to be done that way. But when you come back, so it's like, so things don't work out. So there's a huge mismatch here now. Like I already talked about the taxonomists, people who've been identifying plants based on this thing. I mean, they will not, because they don't want to understand the molecular techniques. They will be scared. They won't discuss. And they are, so when they come to know that the tiny tots, the young, 20-year-olds, they can do better identification than us, it will be very difficult. And so trust me, when you are doing biotechnologies, when you're talking about to the traditional plant breeders, when you're talking about the people from Botanical Survey of India, give them the due respect. Within your heart of heart that they're not outdated, but they need something which they need to catch up with, but they don't have. You can have that, but you have to be very humble. So the point I want to say is that, really speaking, DNA barcoding is only a supporting role. But apart from that, when we go into jobs, when we work with people in industry, people with academia, people who done the identification and analysis using a traditional systems of identification, please, you have to have the right attitude also, and give them the due respect. We have come this far not with the DNA barcoding, not with the molecular technologies, but based on simply eye observations. So the gap is there. Even if you know you're right, sometimes you have to be more humble in dealing with them and giving them the credit. Now, phylogenetics is another thing. Manjunath ma'am, we had a word, and she was very keen that we can discuss phylogenetics. So I have tried to cover up everything in a quick way and with as much detail as was really required by you, and as I thought could be delivered in 1.5 to 2 hours time. And the other thing is, so now we have the time, and tomorrow we can take up phylogenetics, and in phylogenetics we will do tree construction, and then we will do so. I'll send to Manjunath ma'am, I'll send the links to a software called Darwin. Darwin is a diversity analysis platform, and I will give you the data, and we will have a hands-on session. So you will all build, dendrograms, and then we will do the interpretation also. So this whole exercise can be done tomorrow. And phylogenetics is something you cannot do away with. Everybody needs phylogenetics, whether you're doing DNA barcoding, or you're doing molecular markers, or you're doing heterotic grouping, or you are identifying microorganisms, bacteria, or whatever. And phylogenetics is something, pre-conduction, this is something you always need in any, even non-biological areas. You go to politics also, you need to do for phylogenetics. So this is a tool which will be really handy, and we will spend tomorrow's whole day. Tomorrow's 2 hours doing phylogenetics, and I will give the software. I'll give the link to software. This is a free software, and I've been interacting closely with the people who developed this software, from France, from INRA. So I'm pretty much in contact with the team. I've been in touch with them nearly 15, 16 years back. But this Darwin, and that was the time when Darwin was only started, and now 15, 16 years down the line, this has become really famous. And I see publications even from top journals like Nature, where Darwin has been used. So we shall be using this platform. I'll be sending the link to Manjunath ma'am, and then all of you please download and install it. And tomorrow we will start with the data, and we'll do phylogeny. And I hope that will have some, that will be experience. Over to the audience, please. And questions. Okay, right.
Yes, sir. Now, why students are not having asked them to ask at least few questions. They have to ask whether they have understood or not, what part was okay for them. Let us get the comments from them, sir.
Yeah, sure. Yeah. Is
Yeah,
it easy, difficult, what? Anything, please.
yeah,
Yeah.
what they understood,
Yeah.
and then what they want.
Yeah. Okay, brilliant. Okay, thank you. Yeah. Okay, sure. Yeah. So, ma'am, is it okay tomorrow we take up phylogenetics then?
Yes, sir, we can take, but we will start little bit early, sir.
Ma'am, before two.
Huh?
Before two, you mean?
Yeah, before 2.
Why do you want to do that?
Because, sir, after we finish this session, I think valedictory will be there.
All
So
right.
it may
Okay.
prolong. So they may give, means if we call somebody from outside, their speeches will be there.
All right.
So the program may go up to 6 o'clock, 7 o'clock.
All right.
So we'll
Okay.
start a little bit early so that we will complete by 4:30.
Let's start at 1, ma'am. Then.
1 o'clock,
Yeah.
that will do?
Yeah, that's fine. Okay, no problem.
Yes, sir. So that if we complete early, so the valedictory 1 hour will be there.
All right, ma'am. Okay, that's fine. Okay.
So
Okay,
that's
sure.
the reason, sir.
That's fine, ma'am. That's perfectly fine.
Yeah, and I have a question, sir.
Go
Actually,
on.
few students are working on probiotic microorganisms for their project. So they
Yeah.
have identified from different sources. They have got the microorganisms, and they have
Yeah.
cultured, and they have doing the biochemical analysis.
Yeah.
Does DNA barcoding means, can we do the DNA barcoding for these microorganisms? So we have to go for sequencing and then do the
Ma'am,
DNA barcoding.
Ma'am, this is what I'm trying to say. DNA barcoding will do much better than the identifications. Like I told you, I just said that, this lady from Chandigarh University, she's writing a PhD thesis. She couldn't, she did all the biochemical, morphological, she couldn't get it right. So the
Hmm.
identification of microorganisms like bacteria, if you do it with DNA barcoding, is much more reliable. It's much easier.
Hmm. Hmm. So
Yeah,
now what they have, because they are doing their projects on that, sir, and they
yeah,
have
yeah.
done the this one. Now for DNA
Yeah.
barcoding, what is the step they have to do?
You have to come, they have to come to me and get trained.
Yes, then you will give them the training, no, sir?
No problem. I don't know. Yeah, of course, I'll be happy. But the thing is, ma'am, at this moment, yeah, we can think on these lines and we can have training courses. So let people have hands on this thing. And that's not a
Mhm.
problem. I would be very happy. That's what I want to do. Yeah, sure. Yeah.
Okay, sir. So I'll talk to you in detail later on.
Yeah,
What
yeah, yeah.
next can be done,
Yeah,
and
sure.
then regarding the students to be sent abroad and other things. What I have told you, you think on these
Yeah,
lines also.
yeah, yeah, we can talk about that. If students are keen. Okay, ma'am, let me say one thing here. I mean, going abroad, I mean, in this case, and it makes sense that people get exposure and then, it's a bit of picnic and it's also a bit of learning. But let
Hmm.
me tell you that, in India, a lot of things, there's a lot of scope and all those things. To be honest, when I went to Malaysia, in fact, they wanted to send to train their people. My earlier boss, he's a Chinese guy. And
Hmm.
he wanted to send this team. He said, because he says, Rajiv, you won't believe, man. He came right to Chandigarh. He came to Delhi. He says, Rajiv, why did you leave? If you
Hmm.
want a higher salary, why would you anything? Come back to me at any
Hmm.
cost. And then eventually, I said, Chua, I cannot come back now. But if you want to get your student people trained, you can send them to me. So they are sending people to us to get trained, ma'am. And trust me,
Hmm.
all that South Asian countries like Malaysia and Thailand, they need the training from India. So they're coming, in fact, from there to us to get trained. So I'm not sure. I mean, oh yeah, I mean, if you want to go for picnicking and if you want to get some more exposure and it adds to the CV also,
Hmm.
you've been to a different place. So that's perfectly fine. And but apart from that, if you're looking for something which is high tech, you can there's all of it. You can Singapore is a bit different. So in Singapore, you will probably get some of the, but there's nothing in Singapore you won't find even in India. So I mean,
Hmm.
mixing all these things, I think it makes, if you want to mix picnicking and exposure abroad with training and like with training and skill development, then it's, but apart from that, I think it might be a good idea to, within the same budget, you can look within possibility within India and you will get the same thing, man.
Okay.
Yeah.
Anyway, I will talk to you
Okay.
personally, and
No
then
problem.
we'll make things better,
Yeah,
and
sure.
then learn the newer techniques, sir. Okay, then
Absolutely.
tomorrow we'll join at 1 o'clock. You
Okay.
can please send us the link 5 to 10 minutes earlier so that students can join, sir.
Yeah, sure.
I think I missed the earlier part. If the recording is there, then I'll see it to that thing. Because I was little out, so I couldn't see
Okay, so
it.
is it being recorded?
That I do not know, sir. Actually, I am
Okay.
not well, so I am not sure.
Okay. So let me know
So,
also. I think I can also, because I didn't record it. So if you have the recording, please send to me also.
yes, I'll ask the teachers those who are there,
Okay. Okay,
and
ma'am.
then we
Okay.
will see, sir. Okay,
Okay. Okay.
okay, sir.
Okay.
So,
Thank you.
students,
Everybody.
do
Thanks.
you have any single questions? If you have any questions, we'll have the session, or we can. Okay. Okay, thank you, sir. So,
Okay.
thank you very much for today's session and
Thanks,
the
ma'am.
interesting topic of the workshop, that is DNA barcoding. Thank you from all
Okay,
our
ma'am.
sides, sir. Thank
Thank you, ma'am. Thanks a lot.
thank you.
Okay. Thanks. Bye bye.
Okay, sir.
Okay. Thank you, sir. Thank you.